I'm gonna try to tell a little story here, but if there are any questions along the way, feel free to jump up and go ahead and ask at any time. So the way I'm gonna go over this today, so I think we're here for about an hour, is that right? Yeah, so the way I was gonna break up the hour was in a couple of different parts, so there's gonna be a couple of transitions, so don't panic. In the first part, the kind of the biggest part, I'm gonna tell this little story that I like to use to explain the sort of the concepts and the process of genome assembly through this little analogy I like to use. So this is where the Dickens part likes to come in. This will kind of just set some basic terminology, basic concepts, hopefully very understandable. Then the first major transition, I'm going to shift gears from assembly to talk about, well, once we have an assembly, what are some of the things we can do with this? So this is something called whole genome alignment. And that's going to become important because at the end of the talk, when I talk about specific assemblers, specific results, I want you to understand some of the pictures that we're looking at. So I'm going to go over this. So all the slides that I'm going to have are going to be made available on the wiki. I also have tons of resources available on my website. So that's what I would encourage you to do. And at the end, I'll have some sort of summary recommendations, both sort of research recommendations and some of the teaching recommendations on some ideas that I found successful in the past. So the analogy that I like to use starts like this. So imagine you're Charles Dickens and you had just finished writing A Tale of Two Cities, which begins, it was the best of times, it was the worst of times, it was the age of wisdom, it was the age of foolishness. So you're Dickens, you're very proud of this book that you just wrote, your masterpiece, and you want to go sell it, distribute it to the world. So you go to the copy shop and you get it printed up in a very special way. So instead of printing it out, regular pieces of paper, we're going to get it printed up on these very long spools of paper. So hopefully this is my completely transparent metaphor for very long molecules of DNA that we want to go do some analysis on. So Dickens, he's got his copies of his book and he's very excited to go sell it. But on the way out, oops, he trips and it falls into the shredder and it chops it up in this very special way. Again, this is my hopefully very transparent metaphor for DNA sequencing where we just grab these little tiny fragments along the way. So in this example, what's happened is the book, which started out at something like 140,000 words, gets chopped up into about 5 words per fragment. It's in the shredder. All the locality which was originally there is totally lost. You don't know which book you came from, you don't know which chapter, which page, all that sort of locality information, totally lost, just in the shredder bag, all mixed together. So now we have this big problem of, oh my God, I want to sell this, I want to make my fortune. How do we put it all back together? That's the gist of what we really want to do. So if we started with 5 copies, 140,000 words or so, we have about 140,000 fragments in this big shredder bag. The way I like to think of these are like little tiny fortune cookie-sized pieces of paper in there. They're all mixed together, and one thing that's going to become really important, really critical to the whole analysis is that some of the fragments are identical. Now this can happen in 2 ways. So it could be identical because imagine you have the spools, they get chopped up such that on page 1,000 at this particular spot it got chopped so that they're all lined up. But sometimes some of the fragments will be identical because they just happen to have the same words on them. So here I've highlighted in yellow and blue a couple of pairs of fragments that have the exact same words on them, but they actually come from completely different spots in the original book. So of times it was and it was the age of. Notice they have sort of different contexts, but exactly the same words. So what do we do? So one idea is, and indeed this is how all of the early assemblers work. If you've ever heard of FRAP or Tiger Assembler, or Cap3, or bunch of early stuff from the 70s, 80s, and into the 90s, uses a process called greedy genome assembly. So on the left here, I just for our own sort of recollection, is the list of those fragments. I happen to put them into sorted order just because that enables some nice things about looking through it very quickly. But kind of the gist of it is well, let's pick a fragment either at random, or maybe we have some clues about what's a good starting point, or maybe here there's only one fragment that starts with a capital letter, and we want to start at the beginning. In some way, we're gonna pick a fragment to start with. So here I'm going to pick the top of this list. And the whole business about genome assembly, or also de novo transcriptome assembly, is kind of asking the question, well, which fragment comes next, right? If we can just figure out which comes next, and next, and next, and next, and next, and next, we can take this like big bag of shredded up fragments and hopefully piece it back together again into the entire genome. So just to kind of give you some terminology, so this is a fragment. The end of the fragment is sometimes called the suffix, just a fancy word for the end. We kind of want to look through this list here and find some other fragment that begins with the end of this fragment. So technically, we want to find, if this is the suffix, was the best of, we want to find some other fragment whose prefix begins like that. So we can do that very rapidly. Aha, was the best of, hey, times. So now we've taken two fragments that were each 5 words long. We've kind of done some string matching type things, and we found the next fragment. So now we've reconstructed this back into 6 words. That's good. So this would be extremely tedious to do by hand, to do all these little pieces. But that's the sort of thing that computers are very good at, so we can very readily identify what's the next fragment in this way, looking at these sort of overlaps. OK, so we found 2 fragments. We can repeat, find the 3rd fragment, find the 4th. Everything is going well. The metaphor that I will sometimes use is, if you imagine that you had this big bag of those little 4-block Lego bricks. If I had enough of these little Lego bricks, I could build a bridge across the harbor, just piecing them all together. No problem whatsoever. Although interesting things start to happen along the way. So here are 2 fragments that both overlap equally well in the sense that they both have this forward connection there. But it's sort of 2 copies of the same fragment. It's maybe a little bit interesting. No problem, though. It's totally clear that we have, it was the best of times, it was the. But on the next fragment, something very interesting happens. And that's that we start to have our first conflict. Right? So here's going to be 2 fragments that overlap equally well. Times it was the worst, or times it was the age. They overlap equally well. They both match on the first 4 words of those fragments. But those 2 fragments themselves do not agree. They have different words, which indicate we're sort of, if you can imagine, we're snaking through trying to reconstruct the text. We've hit one of these critical branch points where there's some ambiguity along the way. So from the fragments themselves, it's completely ambiguous as to which is next. It was the best of times, it was the worst, it was the best of times, it was the age. There's just not enough information to decide if we're doing this whole analysis sort of one fragment at a time. We need a more global perspective on all these sequence relationships. So we're going to do this using a little bit of formalism. We're going to think about this in what's called a graph context. So the name of the graph is complicated. Is named after an old mathematician whose name was Bruijn. Let me explain it by example, though. So we're going to take these fragments, which started out as being 5 words long, and we're going to do something which at first sounds really crazy, which is to take a 5-word fragment and then turn it into 2 fragments that are even shorter. So we're going to go from one 5-word fragment into 2 sub-fragments, each 4 words, and just keep track of, oh yeah, it was the best of, comes before, was the best of. Now, if we flip back into DNA sequencing or RNA sequencing, this would be something like a 100 base pair Illumina read. And then we're going to chop out from it the first 25 nucleotides, then the next 25 nucleotides, and the next 25, and next. So instead of just having a pairwise relationship, we'll maybe have something like, I don't know, 70 or 80 of these 25 base pair words all connected in a row. This is called building up the Bruijn graph. So this is a super easy thing to do. You just get a fragment, cut out the first 25 characters, next 25, next. Very easy to do. There's a little bit of bookkeeping that has to take place to keep track of which comes first, which comes second. This would be extremely tedious to do by hand, but computers excel at this sort of thing. It gets very complicated when we talk about very large genomes where you need massive supercomputers, but for small problems, this is quite tractable to do so. The trick, though, is in addition to doing this sort of one fragment, one little piece at a time, we're going to do this over the entire collection of fragments that we hope has captured the entire book or the entire genome or the entire transcriptome. So here's me simulating Dickens, taking these fragments, splitting them up, laying them out very carefully, trying to recover how all these different sequences relate to each other. So this is the initial de Bruijn graph that will be formed. And I think here's my proof by picture that we can start to capture a lot more of the global relationships that take place. We can start to see the overall structure of the original book or of the original genome or the original transcriptome. So notice when the graph is first constructed, every sort of node in the graph, every little sequence that we're going to consider is exactly the same length. In this example, every piece here is 4 words long. There are sort of relationships between them. Almost every node has sort of a successor. Almost every node has one successor, sometimes 2 successors, and so forth. Parts of this graph are very simple. it was the best of time. There's sort of no conflicts along the way. It's very clear what the sequence has to be. Sometimes there's conflicts along the way. The goal of what we want to accomplish is sort of take all this global information. Again, this is just a tiny snapshot into Tale of Two Cities. Take all this global information and try to reconstruct the entire book. So the computer, the software will build such a graph, and then a natural thing to do is try to simplify it, try to look for what are the segments that are easier or more difficult to reconstruct. So a natural thing to do is to look for parts of the graph where there's no conflicts, where there's no ambiguity, and basically put them all together, put them into bigger and bigger pieces. So here's me simulating this process. Aha! Now I've taken this graph that started out where every node was exactly 4 words long, and now we have some sort of variable length sequence up to where these branches occur, in particular because of repeats. So given this graph, I think it should be clear to everyone that the book has to start kind of over here because there's no incoming edges. It has to end over here because there's sort of this one dangling edge. So the concept is that we want to find some path through this graph that starts over here, ends over here. This is a very simple graph, and you might be tempted to say, oh, well, it has to start here, walk through there, down here, out there. And that's because in English, it is extremely awkward to write. It is the best of times. It is the worst 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 of times. It was the age of wisdom, 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 age of fools. It would be extremely awkward to write that in English. But of course, as we start talking about real genomes, that sort of tandem repeat structure can occur all the time. So we have to be extremely careful over what sort of simplifications that we want to apply. And even more problematic, this is a tiny snapshot. As you start to look at more complicated sets of sequences, so here, rather than just taking the first sentence of Tale of Two Cities, we start looking at this sort of sentence structure across the whole book. For whatever reason, Dickens liked this sort of structure to the text, so you start to build up a much more complicated graph. So this is the mental model you should have whenever you have a genome assembly or a transcriptome assembly. There's going to be this incredibly complicated graph. If we get lucky, some parts of it will be linear, will be straightforward. Those will represent our initial contigs where we have a lot of confidence in them. But then there's going to be tons of branch points, tons of relationships because of, in particular, because of repeats and because of errors in the data, because of heterozygosity or diversity in the sample, or sort of any other sort of technical biases along the way. So it's actually a hard problem in that sense. Now, given one of these graph structures, there's lots of ways that you may want to try to summarize it. Maybe we're going to try to pick out maybe the longest node or some sort of average about it or something like that. But in practice, the way, my slides are a little bit out of order. Sorry about that. Let's go ahead and complete that thought. So the way that we're going to think about this, I took out that slide completely. That's why. Sorry about that. So the way that we're going to think about this is to compute some sort of average over these sequences that will represent the entire assembly. We'll come back to that point later on. So that's sort of the concepts behind genome assembly or de novo transcriptome assembly. Let's think a little bit more detail about what it means to be a repeat. So here I have a cartoon version of a genome. It's a sort of trivial. Has some, and instead of labeling individual nucleotides, here I just have segments labeled. So here's a unique segment I'm going to call A, then a repetitive sequence I'm going to call R. Notice there's 3 copies of it, then B and C. Now, if we simulate some sort of perfect shotgun sequencing of this genome, we're going to get all these short fragments along the way. The critical thing by short and long is all sort of relative, right? If all of our reads were 1 or 2 base pairs long, of course everything is repetitive. On the other extreme, if we could read off entire chromosomes, nothing would be repetitive, right? We get the right answer. So there's this gradient in between where being repetitive or being in terms of the assembly graph is entirely relative of the length of the read relative to the length of the repetitive segments. So here we have short reads that are shorter than the repeats that are present. Now, from the point of view of the assembler, it's going to look at all those relationships and build up some sort of graph. So here's probably something like the graph that it'll construct, where it's going to start in this A segment, walk into this repeat R segment, then have this branch. You don't need to do anything special about repeats for the assembler, right? It'll readily detect that this is the pattern here. But then really the key point is there's this uncertainty over how things are connected. So in principle, all the information in the assembly, in the genome, in the transcriptome is present. It's just not clear about how it should be represented. So from this assembly, the assembler would probably output 4 different contigs representing these 4 different segments along the way, which depending on the type of analysis you're doing, that could be okay. But if you want sort of global analysis of the chromosomes or of individual transcripts, we need to add in some sort of extra information. In particular, the information that we need is something that can link basically from one unique segment to the next unique segment to the next unique segment. One way to do that is through longer read technology. So at the end of the talk, I'll give you a little snapshot into some of the PacBio sequencing that's available that can generate, instead of reads that are on the order of, say, 100 base pairs, we're now pretty consistently getting reads over 10,000 base pairs, which just gives us much better information for linking together segments. From the point of view of the assembler, what that does is, if you start with some sort of complicated graph like this, you can think about taking these long reads and sort of squinting at it and be like, oh, well, it starts in this A, and then it goes through this repeat somehow, it comes out on B. That's, that sort of analysis is exactly what happens inside the software. And then suddenly the assembler can be like, oh, well, yes, from the short reads, there is this complicated 4-node graph. But if we can sort of thread through there the paths traced out by these longer reads, we can identify there is a unique path through there. And then as many copies are needed, or as indicated by the long reads, we can create those repetitive nodes and then hopefully generate an entire genome end to end as a result of that. This is a good day. When we started from whole genome shotgun sequencing and we're able to assemble up, in this case, the entire genome or the entire transcript. Today that is only possible, in practice in microbes, in small eukaryotes like fungi, and also it's only possible using the current long read technology. You're just absolutely not going to take whole genome shotgun data from a mammalian genome and get entire chromosomes. This is not going to happen. Instead, you're going to get a snapshot of many thousands or many millions of contigs representing different portions of those chromosomes. And there, you'll probably have to do some sort of post-analysis where we're going to use maybe longer reads or May pairs or optical maps or genetic maps, some sort of other information to be able to take those short contigs and then lay them out into longer and longer segments. This is definitely the limit to the research, and today it's mostly a limitation in the biotechnology. We more or less know what the right algorithms are, but the challenge is incorporating these new biotechnologies that have weird error rates or weird biases, or what's the right combination of technologies to bring them together. Okay, so that's hopefully the very understandable introduction to genome assembly. Let's sort of transition now into the real data. So genome assembly has a very long history in genomics. It's really fundamental, right? So the very first sort of complete organism published by Sanger when he was publishing the Sanger sequencing paper, for which who won the Nobel Prize with, he was not able, even then, he was not able to sequence 5,000 base pairs in a row. He was able to sequence little tiny fragments that then had to be reconstructed into larger and larger segments. In 1995, at the Institute for Genomic Research, where I used to work, we published the first free living organism. This is of Haemophila influenzae. It's about not quite 2 megabases long. Really, the milestone there was, again, we sort of knew how to do it from this, but we were able to ramp it up by a few orders of magnitude to go to a much larger genome, to be able to fully capture an entire genome for the first time. And then throughout the 90s, this got continued to be optimized in terms of the biotechnology, in terms of the algorithms, and we saw for the first time the first large multicellular organism. We saw C. elegans. In 2000, we saw the first large genome published using what is now commonly called whole genome shotgun sequencing, whereas C. elegans was published in a so-called back-by-back way where you try to localize the analysis. In 2001, we saw the publication in the Human Genome Project, both the private and public efforts. And then I would say that the next really critical milestone was in about 2010 when we saw the heyday for Illumina sequencing, and we saw the first mammalian genome that was ever sequenced using short read technology. So it was really really high profile scientifically on the front cover of Nature. As we look at that assembly today, we've come to realize it had some problems. It was very early Illumina data. The reads were very short, and as a result of this, the assembly was really fragmented. So now there's efforts to go back and try to improve upon it. So I guess that's another conclusion from this talk is genome assembly is never done, right? We're always going back, trying to polish, trying to incorporate new technologies, new ideas, and make it better and better and better. So the applications for these algorithms, for these ideas, are pretty widespread. The most natural application are applying to novel genomes. You're really interested. Just today I got an email. We're really interested in this particular butterfly. There's no reference genome available for it. It has all these amazing properties. We really want to understand evolutionary. We want to understand all the regulatory elements. We want to be able to really map out all these traits. Can you please help? I get this email not quite every day, but every other day or so. And I'm happy to collaborate with people, although I can't assemble every genome on the planet, but so be it. So that happens sort of in individual investigator labs all the time, and then also there are these bigger projects trying to look at more genomes at once, right? So there's a big effort launched by David Hostler and friends over in UC Santa Cruz to sequence 10,000 vertebrate animals. I'm teasing a little bit, but the spirit of the project is let's go to the San Diego Zoo and yep, we're just going to sequence everything here. Scientifically, the rationale for this is one of the most powerful ways that we have to understand the human genome or any genome is to compare it to sort of to compare it across time in terms of the evolutionary forces that are present. So if we see some sequence that's been really highly conserved in humans and primates and other mammals and chickens and across the tree of life, that can be a really good clue that this is significant. Conversely, if we see that there's been a sequence that's been really well conserved from in the other way, from fungi to other vertebrates to mammals to primates, and then suddenly has been rapidly accelerated in human, this can give us some good insight into what is the sort of genomic basis of some uniquely human traits. So he's not here today, but Adam Siepel at Cornell, who's going to be the newest member of the faculty here in quantitative biology, this is exactly the type of question he asked, has identified these sets of, he calls them human accelerated regions. So regions where there's been really an exceptional amount of evolution taking place in genes that seem to be associated with uniquely human traits. Really interesting, really cool stuff. So that's 10,000 vertebrate species. There's thousands of insects, plants, microbes. Really, literally across the tree of life. There's big efforts underway to establish those genomes. In addition to one genome at a time, there's a very closely related problem that of called metagenomics. So the idea there is we're going to go to some environment, and rather than trying to isolate and sequence one genome, we're going to sequence many. The entire composition of all the species that are present there. So a really compelling example of this is the Human Microbiome Project. This is what this logo is, where we're organized through NIH, where they've been sampling on many different body sites, some that are obvious, skin, mouth, hair, some kind of gross ones. And they're just sort of asking the questions, what is the diversity present? What are the relationships to sort of typical diversity to stress environments, disease environments, things like that. A major conclusion from that type of work has been diversity is good. If you have an infection, it can really take over. That's when you start to have problems. And then another major conclusion from this has been there's been some really interesting studies where they've taken skinny mice and obese mice and swapped the stomach contents in a very gross way. But the outcome from this is that the skinny mice become obese, the obese mice become skinny with no other changes to diet or anything like that, exercise. So it really sort of demonstrates that these microbes play an extremely important role in our own metabolic processing. So it's very interesting to study them. I feel like it's a really open frontier over, in which scenarios are the microbes causal, and in which scenarios are the microbes reacting to some other fundamental change. I think there's a lot of open questions there, but it's an amazing field of research. Another example of this has been, gotta love the guy, Craig Ventnor, sailing around the world, collecting the seawater to filter out all the different microbes that live there. So this is the logo from the Global Ocean Survey. At the time that this was done, they expanded the universe of known genes by something. It was an incredible amount, like a factor of 10 or a factor of 100, just because there is so much diversity of life on the planet that we just don't ever encounter. The idea for this is, once we build up these big catalogs, we'll just have so much better understanding of evolution, biological processes, opportunities to discover new metabolites, new interactions. It's just amazing about the things that we can do. In addition to sequencing genomes or metagenomes, these assembly techniques are now becoming a really fundamental part of many different sequencing and biological assays. So I know the spirit of the class here has been transcriptome analysis. And yes, you can take RNA-seq reads and map them to a genome, but there's also a sort of a parallel line of thought where we're going to be taking those reads, comparing them to each other, trying to assemble the transcripts de novo. There are various pros and cons of both approaches. And I think that's true. That'll be true for a long time, where in certain scenarios it makes sense to map, in other scenarios it makes sense to assemble. As the reads get longer and longer and longer, though, I think we're going to see a bigger and bigger shift into doing some de novo assembly up front, just because it's you sort of free yourself from any sort of biases over what may or may not be present in the reference genome, especially considering that the reference genome is a misnomer, right? It's really either Craig Ventnor's personal genome or the individuals that led into the public effort. So in addition to sort of transcriptome analysis, there's a lot of exciting work where you're going to use assembly techniques to look for genetic variations. So BGI had a nice paper where they're interested in structural variations in humans. So rather than trying to map this out through read mapping, they assembled that individual, did the mapping at that level. In my own lab, we have algorithms. We're interested in particular in indels, so insertions and deletions of more than a few base pairs. Works really well to do an assembly up front and then compare those contigs back to the reference rather than trying to figure this out through mapping directly. So the process is exactly what we just saw with Charles Dickens, except that instead of physically going through the paper shredder, we're going to shear and sequence our DNA into short fragments. Again, the whole business of genome assembly is detecting and sort of analyzing these patterns of overlaps where one read matches the beginning of the next to the next to the next to the next to the next. The graphs that are generated are very complicated. You're certainly not going to be able to assemble a million-sized genome on a laptop. That's why sort of 3 floors below us is where our data center is. The biggest computer in that room has 1.5 terabytes of RAM. It's legitimately a supercomputer. And sometimes my lab does run that computer out of memory, which case I have friends at Pittsburgh Supercomputing Center where there's a machine called Blacklight that has 32 terabytes of RAM. As far as I know, this is the biggest shared memory machine on the planet, and I occasionally run that machine out of RAM as well. So it's just sort of operationally and computationally, there are some extreme challenges of scale. There's some very clever algorithms that are being developed that are trying to, as we go to larger and larger genomes or larger and larger metagenomes, with more money, you can scale up to a limit, but then at some point, you just need cleverer and cleverer algorithms to reduce the space requirements. As the graphs are constructed, then there's, like we saw before, efforts to try to simplify them down. This will define your initial set of contigs. So if you've ever run, for transcriptome assembly, if you've ever run Trinity, this is exactly what the first component inchworm does, right? It's going to build up this de Bruijn graph, try to put together these sequences that are where there's no ambiguity. And then we're going to take this complicated graph, try to detangle it using longer reads, mate pairs, genetic markers. You can sometimes get away with using synteny, but you have to be extremely careful, right? If the conclusion of your paper is, we see that the gene order is really well conserved, but you've used synteny to determine that gene order, these circular arguments can get you in trouble with reviewers and editors sometimes. So we've hinted at this, but let me kind of spell out why it is hard to assemble a genome. So of which there's many reasons. So biologically, in practice, we're not just assembling one book. We're actually assembling, in the case of human, 2 books at the same time. If we're scaling up to, say, wheat, we're actually assembling 6 books at the same time. If we're assembling, strawberry, there's 12 books at the same time. As we're going to some of these more exotic plant species, we're talking about dozens of books at the same time, all of which have differences between them. So we have ploidy and heterozygosity can be very complicated. The big issue, of course, though, is repeat content. Even if it's a haploid human genome, you're going to have those repeats where all the transposons in particular have been jumping around. It's going to be in segmental duplication. It's going to be very complicated to put those together. At the level of sort of technology and biotechnology, they're very large. It can be very expensive to generate data for that. Fortunately, especially Illumina technology is rapidly decreasing in cost, such that it's becoming more and more affordable to go for these larger genomes. The instruments make mistakes along the way, though, so we have to be careful about sequencing errors and lack of coverage because of weird motifs or just other sort of systematic errors in the data that come along there. I've talked a little bit about sort of the computational challenges along the way. You're not going to be able to do it on a laptop for large genomes, just full stop. There are efforts underway to improve the algorithms. There's access to larger supercomputers or scientific computing centers that you can use, but that's a big problem there. The one that I'm not going to have any time to talk about, unfortunately, is how do you assess that you have a correct assembly at the end of the day? There's a lot of research into this. If you're interested in this, if this is what you're working on, I encourage you to check out the Assemblathon papers. So this was this international effort to take a data set, get all the leading assembly groups around the world to assemble the same data set, and then ask the question, how do they relate to each other? I think a major outcome from that is there's no perfect assembler, and then the corollary to this is that it's very complicated to assess a de novo assembly to say if it's correct or not, unless it's from simulation. But in those projects, we've described some metrics that can be used to try to say, yep, this looks good, this looks bad. Size in itself is not a great indicator, right? You can trivially sort of smash things together if you want big size. So you have to be a little bit cautious over just trying to say, oh, my contigs are this big or that big. That is not necessarily a good indicator. So if you want a good assembly, there's certain ingredients that you just absolutely have to have, or otherwise your assembly is doomed. So the number one ingredient that you have to have is sufficient coverage, and we'll see a few more details about that in a second. But the idea is we're doing this sampling from across the genome, and if you have 1x coverage of your genome, you do not actually have one full complete copy of the genome. You have to oversample considerably to build up enough coverage so that you do have that good sampling. So very fundamentally, that's the first thing you need to have. The second thing you need to have is sufficient read length. So what I'm trying to show here is, imagine we've done, Illumina sequencing. We've built up one of these de Bruijn graphs using your favorite assembler. It can detect where these repeats are, but assemblers tend to be pretty conservative. Rather than sort of smashing things together, making mistakes. Maybe this is a part of chromosome 1. This is a part of chromosome 2. From the point of view of the assembler, it doesn't know that, right? So rather than forming chimeric contigs that start on chromosome 1 and jump to chromosome 2, in particular because of repeats, it'll probably output multiple contigs just to be safe there. But if we can overlay that information using mate pairs or longer reads, we can say, aha, these contigs came from chromosome 1 or some chromosome. These contigs came from some other chromosome. Let's separate them out. Let's resolve across those repetitive elements. Again, it's all relative. The lengths that you need for microbes are very different than you need for mammals or pine trees, just because those larger genomes tend to have much more complicated structure to them. And then the third thing that you need is just really high quality data. If there's lots of errors in there up front, it's going to be very difficult or impossible to detect which reads overlap with which other reads. My lab and other labs spend a lot of our time developing new algorithms that can try to overcome some of the errors in the data. But very fundamentally, if you have bad data going in, you will get a bad assembly on the way out. It's just the reality of it all. So did you guys talk about Illumina sequencing? OK, so the vast majority of the data that you see today will come through this pipeline. Since you've already talked about it, I won't go into any great detail, other than to say that the output from all of this are going to be these enormous text files that just have gazillions of these short sequences that are present there. So the only the most tractable parameter to control in a sequencing project is how much sequencing to do. Do I do, in practice, do I do 1 lane? Do I do 2 lanes? Do I do 10 lanes? How much should I do here? Again, because it's this randomized process, conceptually what you would want is, oh, well, I want just enough such that the reads will just perfectly lay across my chromosomes. That'd be amazing, but you get no such guarantees. So the mental model that I like to have for this is, imagine you're out in the street. It's a dry day, but out of the blue, oh my God, there's a freak rainstorm. So the first raindrop that falls on the sidewalk will be in isolation. It'll land there. It'll form a wet spot. Next wet spot. The next wet spot. Next wet spot. And those first few raindrops will hardly ever overlap, right? They're hardly ever going to touch each other. It's not until it starts to saturate the pavement, then suddenly what started out as isolated raindrops will start to form little tiny puddles, and then finally, with enough and enough and enough, we're going to have to finally be able to cover the whole surface. Now, the amount of rain that has to fall in terms of surface area is much larger than the actual surface area of the sidewalk, because it's this random process. Just the probabilities are against you that the raindrops are going to fall in a nice uniform way that you want them. The only way to get uniformity is to do so much extra that you can pick out the critical reads that you need. So in particular, here's my in silico experiment where I have say 1,000 base pair genome, where I'm dropping in reads where I know exactly where every read goes. If I do 1x coverage of the genome, we get some profile that looks like this. Where yes, on average we have 1x sampling of the genome. But sometimes we have a bit more. More problematic is sometimes we have less. So here in the corner is a little histogram. The sort of takeaway from this is if we do just 1x sequencing, we're actually going to be missing at least 40% of your genome. Just the way the probabilities work against you, you're just going to be missing a lot of data there. If we double the coverage, on average now we're up to 2x coverage. That's good, but we're still going to be missing quite a bit of the data, something like 15% or so. If we boost it up to, say, 4x coverage, things get a little bit better. As we boost it up to 8x coverage, finally we reach the point where we hope the vast majority of your genome will be covered. Back in the Sanger days, you lived and died by 8x coverage because that was the point where the statistics were in your favor, where you hoped that you had enough coverage such that 99.9% of the genome would be well covered. That's great in terms of a percentage, but 0.1% of the human genome is still megabases of stuff that's been missing. So that if you want to actually get full composition, you better go deeper than an 8x coverage, plus the data have errors, plus there's biases. So my recommendation today for Illumina sequencing is to target 100x coverage, just so that you have good sampling across the genome. As you go up to higher ploidy and other sort of complicated environments, things get really complicated where you may need to go far beyond this. If you're talking about transcriptome work, right, the hard part there is the high dynamic range. So if you need at minimum, say 10x of those low abundant transcripts, now you're talking about hundreds or thousands of x of those highly abundant transcripts. And it's totally driven by this. By the way, this little distribution here is a mechanistic way to get at the Poisson distribution. So that's exactly what this is. So I think I just said all of this. There's this nice article here that summarizes those results. Okay, so we can do all the sequencing. From that, you can sort of, you can do some estimates over genome size. You can work backwards and be like, oh, I need, 1 lane, 2 lanes, 3 lanes. From that, we can load it into the assembler. In 5 minutes, I'll tell you a little about those assemblers that are available. Inside the assemblers, it'll create one of these big assembly graphs. It'll try to simplify that. Again, there's, I have 3 in there twice. There's 3 or 4 different ways that you can possibly come to have sort of branches or gaps along the way in the assembler. The easiest ones to deal with are, actually the easiest one to deal with is coverage, right? Because then you just pay more money, get deeper coverage, hopefully life will be good. The next easiest is random sequencing error along the way, right? So here's me. This is a mental model of an assembly graph, actually from a very small microbe. If there's just a little sequencing error along the way where, instead of an I, there's a Y, the assembler will sort of self-discover this in the sense that it'll lead to these little patterns in the data where this is called a little tip that'll be hanging out, or if there's either there's heterozygosity or there's just the wrong sequencing error, you can get these little bubble motifs. Fortunately, though, the assemblers are very competent at detecting these patterns and trying to smooth them out, assuming that it's not crazily complicated, in which case you would need even more sophisticated algorithms. So given those data, the assembler will build a graph, correct the errors, try to put it together into these longer and longer initial contigs. At the end of the day, though, the limiting factor will be the repeats, right? This is where all complexity in genome assembly comes from. This is where all complexity in a lot of the complexity in transcriptome assembly comes from. This is actually also where all of the complexity in variant detection or trying to map out, I don't know, ChIP-seq peaks or any sort of genomic analysis is all the repeats that cause all the problems to a first approximation. In mammalian genomes, there are many different categories of repeats. So the conclusion of this is expect at least half of your genome to be hard to work with. If you're lucky, sometimes it's a lot worse than that. Again, from the point of view of the assembler, it will readily detect where these repeats are. So again, if this is our mental model of perfect shotgun sequencing of this genome with a 2-copy repeat, from the point of view of the assembler, what it will see will be something like this, where there'll be initial contigs representing unique segments, initial contigs representing repeats. This is all by design to sort of smash the repeats together up front. It can then notice, oh, well, the repeats have twice as much coverage as the rest of the genome, so that's where the repeats are. So more formally, there's some statistics that can apply to this, try to detect that. So the reason I bring this up is you don't, using sort of modern tools, you don't need to do any repeat masking up front. That was very important back in the day, but now the assemblers are very capable of detecting repeats on their own using these various statistical tests. And then once we have that, the next part, like we mentioned, is if we want to take short contigs and then link them up together into nice big scaffolds, that's where we're going to need to overlay other types of information. Mona, did you talk about sort of mate pairs and paired-end sequencing? Yeah. Yeah, OK. So this is a way where in addition to getting contiguous sequences, we can get this so-called linking information where you'll have pairs of sequences that are separated by approximately a known distance. The reason why you might want to use this is that, say we had some initial contig here, and we knew that its mate pairs were supposed to be, 10,000 base pairs away. We can build up these scaffolds that will link together those initial contigs. The challenge for that is you won't necessarily know what's in the middle. So the way that the algorithms work is build up these initial scaffolds out of things that we have a lot of confidence in. That's what I'm trying to highlight in green. And then go back and try to place some of those more repetitive initial contigs along the way based on where those mate pair relationships are determined. It works well, but like I said before, you hardly ever or never will see perfect genome assemblies come right out of the assembler. In practice, what you're going to get is going to be a mix of some very big contigs and some smaller contigs. The way that that's most commonly described today is using this metric called the N50 size. So the N50 size says that half of your contigs have been assembled into this size or larger. So the way that this is computed is, say we have a 1 mega base pair genome. We have a very big contig, 300 KB. We have some intermediate ones. We have some little guys. You could imagine computing the mean of this or taking the median or whatnot. But for whatever reason, the way that we're going to look at this is through this N50 size. So we'll just put them in sorted order from biggest to largest. The biggest doesn't yet reach the halfway point. This one doesn't reach the halfway point. Oh, it's only when you get to this contig here that's 30 KB that you've reached the halfway point. So that's the N50 size for your assembly. This is the most common sort of scenario where you get a few very big ones and this long tail, little teeny tiny ones. These all represent very complicated repeat structures or any residual error in the data. This is always what you get. So, if all things being equal, comparing two assemblies to each other, you definitely want a larger N50. Although, like I said before, if people have been trying to, optimize for it, this is an easy metric to cheat because you can just smash contigs together. Have to be a little bit careful. Okay, so I'm running about 20 minutes behind schedule. So we have a critical question. I can jump ahead to some of the actual assemblers. I can talk about the current research, or I can give you some sort of summary and some recommendation, but I cannot do all three. I think I will. Mona, do you have any insight here? Yeah. Current research? Okay, good choice. Good choice. So again, all these slides will be online. Actually, I did a other videos that you can go check out. OK. So current research is talk about long-read assembly. So I've been, in a funny way, so when I started at Tiger a million years ago, the hottest technology on the planet was this algorithm called the Solaris assembler. It peaked in popularity as part of Drosophila, as part of the human genome, part of mouse, dog, many of the mammals that were done at that time. Until about 2005, when 454 came along, and then Illumina, and then it kind of really waned as the new technologies came on board. And that's because it was designed around 1,000 base pair reads, more or less, but it stinks at 100 base pairs. So if you have an Illumina-only assembly, you should not use the Solaris assembler. The one I recommend is called AllPass, produced by the Broad. They've done a great job making it relatively, actually very easy to run and operate. You generally get a very good assembly as output. I don't know if there are iPlant users in the room, but we've installed it in the discovery environment, so it's relatively straightforward to run Illumina-only assemblies in there. The one critical component of AllPass is that you have to have two libraries. You have to have a fragment library where the reads actually overlap with each other. And that's really fundamental to the algorithm. You can do various hacks to try to get it to run without that, but you'll have a bad assembly. So you're better off designing your experiment up front such that you have this so-called overlapping pairs library. You need that, and then you need one jumping library. Things will be generally pretty good. Your expectation should be contigs on the order of tens of KB. If you have really nice jumping libraries, then your scaffolds will be maybe hundreds of KBs, megabases if you're really lucky. But that's the reality of what you're going to get in a simple environment. Sometimes much, much, much worse, right? So in Nature a few months ago, there's a publication of the wheat genome. In reality, what was assembled were these very short contigs that averaged, N50 size was like 2 KB, right? So it's not actually anything resembling chromosomes. It's these little 2 KB fragments. That's the state of the art with short read sequencing. So if your project really requires bigger than that, that's where we need to look at long read technology to take it to the next level. The way that works is very similar to what I described with Dickens, right? It's going to compare the reads to each other, compute overlaps, simplify the graph that is derived from that. The special thing, though, is that it's really been optimized for long read sequencing technology recently, right? This has all been ongoing over the last 15 years. Today, there are 3 long-read sequencing technologies. I think you guys on Wednesday had a really nice introduction to PacBio sequencing. So that's the main workhorse that we have in our lab in Cold Spring Harbor for long-read sequencing. And it's all driven by this picture right here. So this is a distribution on the read lengths that we're starting to get from these instruments. So the units here are tens of thousands of KB. So this is 10 KB, 20 KB, 30 KB, 40 KB. The longest read that we've ever had on the instruments here have been 54,000 base pairs. The longest read on the planet, I believe, was generated just down the road at Mount Sinai is like 84,000 base pairs, right? So this is like huge, huge, huge pieces of DNA that are starting to be generated by these instruments. The challenge for today it has a high error rate, but we more or less know how to deal with that through software. And also a relatively low throughput compared to, say, Illumina sequencing, such that it's going to take quite a lot of money to sequence a mammal today to deep coverage. The good news is if you're interested in microbes or yeast or smaller vertebrates, there it's affordable to do so. It's similar in cost to, say, the corresponding jumping libraries that you would need. So it's probably a good idea to go for that. Just so you're aware of it, there are some competitors to PacBio for long-read sequencing. There's a technology that was recently purchased from BioLumina called molecular technology. In the interest of time, I just can't go through it, but the gist of it is a way to generate fragments that are on the order of 5 to 10 KB. The virtue of those data is that they're basically perfect. So it'll be like perfect 10 KB read, a perfect 5 KB read. The challenge with it is just fundamental to the way the technology is derived. It's going to be very hard for them to take it much further than this. So it's sort of a great step up from high-seq technology, but it's talking to very smart people. It's going to be really hard to push that up any higher. The very new technology that's just starting to be announced is this nanopore technology from this company, Oxford Nanopore. The nice part of this today is that it's extremely mobile, right? It's a thumb-sized device. You can walk around. You can take it to remote labs. I guess the DNA, where's Jason? The DNA Learning Center has a couple. I saw you tweeting at a high school with some people that have this. Yeah, yeah. So that's super cool that we can go do that. The people are reporting relatively nice read lengths on the order of a few KB. So this is my visual recollection of what was presented at this meeting in February. You get a nice couple KB reads out of this. The challenge, what people are starting to report, is very high error rate. So just yesterday there was a report of a 30% error rate from these data. It's going to be challenging for a while to figure out how we're going to use these effectively, but super cool for the future. Potentially, very scalable, right? We can go from thumb-sized devices to many of these, and actually that's their product line, is to scale it up to these bigger instruments. The challenge for this is, even with PacBio, it's the very high error rate compared to Illumina sequencing. Depending on the run and the day of the week, it's between 10 and 20% error. So my lab and other labs around the world, though, have developed different algorithms depending over, and really the critical question is how much coverage do you have? If you have very deep coverage, 50x or beyond, the right thing to do is PacBio has a pipeline called HGAP that takes those reads, even in their high error rate, compares them to each other, and then is able to do like a little on-the-fly assembly of that, compute the consensus, and that becomes your error-corrected read. So now you more or less have perfect reads that are 10,000 base pairs, 20,000 base pairs. That's great for both genome assembly and transcriptome analysis, where you just get this beautiful data as a result. So just to kind of give you an example, so this is sequencing we did here at Cold Spring Harbor. Well, these guys in the back did here at Cold Spring Harbor back in February. So this was an afternoon's worth of sequencing. We generated 275x coverage of yeast of Pombe. Kind of a couple key metrics from this data were mean read length was 5,000 base pairs. We had over 100x coverage of reads that are 10 KB long or greater. We even had almost 8x coverage of reads that are 20 KB or longer. Max in this data was 35 KB. So we ran the error correction pipeline. We then assemble it using the Solaris assembler. This is called a dot plot, showing the results of our assembly against the reference Pombe sample. So on the y-axis here are my sort of contigs laid out end to end. Notice there's just a few of them. There's kind of one big one, smaller one, smaller one, a smaller one, and then another big one. Over here we have the reference genome, and there's actually some breaks along the way here, and that's because this is chromosome 1, chromosome 2, chromosome 3, and the mitochondria. So we basically had one contig representing each chromosome of Pombe, which is an incredibly amazing result, considering that 10 years ago it was a many $million-dollar, many-year project to sequence one microbe, and now in an afternoon we can generate all these data. We get more or less a perfect assembly as a result of that. So yeast is about 12 megabases. Oh, the only place where the assembly was not perfect was in the presence of a very complicated repeat that just the reads weren't quite long enough to span across. As we scale up an order of magnitude up to Arabidopsis, PacBio generously sequenced and made publicly available this very nice data set where it was over 100x coverage in Arabidopsis. When we take those data and we assemble in our own hands, we assemble it into contigs whose N50 size are 8.4 megabases. So again, in my mind, this is an amazing result. We're more or less assembling entire chromosome arms from Arabidopsis into individual contigs. We have incredible power to study all the genes, all the regulatory features, do all this amazing comparative analysis. It's light years ahead of what you would get from Illumina sequencing. Just to put it into context, so we did an assembly using what's called a MiSeq instrument, which generates 250 base pair reads. Instead of getting contigs that are megabases, we get contigs that are a few tens of KB. The crazy thing is this is state of the art. This would probably be a publishable result. Assuming there's enough sort of research into this, this would probably be a publishable result on its own. And yet here we are, light years ahead of what you can do with Illumina sequencing, all driven by the long read lengths that are possible. As you scale it up again to the human genome, this is data that was also generated at PacBio in collaboration with WashU, where they had sequenced what's called a haploid human genome using kind of the latest technologies. They generated over 50x coverage from this. And then the incredible thing from this, again, is we're starting to see the contigs being generated on the order of several megabases. Now, because human chromosomes are so much longer, this is not yet individual chromosome arms by any means, but it's showing the path forward. Just recently, PacBio has announced they have in-house, they have some technology that's going to extend the read lengths, improve the throughput, and then suddenly everything, knock on wood, should link together even better and better over time. So just to sort of summarize, so assembly quality very much depends on coverage. Your assembly and your transcriptome analysis is mathematically doomed if you do not have good coverage. The repeat composition of either the genome or the transcriptome, sort of the read lengths that you have present there, it's very much a relative relationship. The whole process is incredibly hierarchical, right? We're going to start from individual reads, look at overlaps, build up initial contigs, overlay it with mate pairs, overlay it with genetic maps, other sort of long-range information to try to put together entire chromosomes. You just have to be really careful to watch out for sort of all these complicated things. If you're trying to publish this, please call me up, right? I'll talk to you about strategies that we can use to try to help you detect that we're not making too many mistakes along the way. In terms of specific recommendations, if you really want to assemble today a good genome, if you can afford it, the right thing to do is deep coverage of PacBio sequencing. As you move to larger genomes, this becomes extremely expensive, right? So now we're going to try to balance between PacBio coverage and Illumina coverage. That's exactly the problem that my lab works on. We're starting to see really incredible results using that combination of technologies. And then because there's a lot of educators in the room, one really effective thing as a training exercise, one thing that I've set up, and you can read about in this paper here, is a year and a half ago was the 60th anniversary of the publication of The Structure of DNA. So we ran this fun little contest where we set up these different genomics challenges to try to teach people about genomic analysis, and part of this was genome assembly. So what myself did and a friend of mine, James Taylor, did is we prepared this sort of contest where you had to decode a secret message that we embedded into a genome. So we took a genome, we prepared this secret message, we put it in there, we simulated shotgun sequencing of these data, and we just put it out to the world. And it's basically the first person in the world that could decode the secret message won some prizes. There's a paper that describes how we did that, and in fact, if you go to GitHub, all the source code to generate the test data and also solve the challenge are available there. I found it to be an extremely effective and also kind of fun way to teach someone the logistics of how you assemble a genome. You know when you have a right answer when it's like, oh my God, there's this secret message that I just discovered that was buried inside of there. It's very satisfying when you come to learn these things and are able to accomplish that. So of course I don't do all this work by myself. I have a great lab of students and postdocs, great lab here at Cold Spring Harbor of amazing collaborators and really supportive funding sources that support all of this research. I will thank you and take any questions, and then just, again, all of these materials are on my website. Please use them, please steal them. That's why I put them out there for you guys. So thank you very much.
Is it possible to kind of combine both, like the long read PacBio and do it at some like a low coverage in terms of to be economical, and then fill in with like shorter reads?
Yeah, so that's very much the research frontier is what's the right way to put these different data together. So you have a few options, right? So if you have a pretty good Illumina assembly to start and you just want to improve upon, close the scaffold gaps, there's a nice pipeline from Baylor called PB Jelly's kind of play on PacBio. So PB Jelly does exactly that, where you have scaffolds, there's a gap in the middle, you'll try to map relatively low coverage of PacBio reads to fill in the spacing there. And then once you have any sort of even rough draft of a sequence, then you can then go back, map your Illumina data to it to try to polish it up. So that's quite practical at below 10x coverage of say PacBio reads. From 10x coverage to about 50x coverage, the right thing to do is call me, because my lab has an algorithm that any day now will be submitted for publication. where we can take the Illumina reads, do some pre-processing, and kind of the gist of it is to take those Illumina reads, map them to these erroneous PacBio reads, do error correction of those, and then from that assemble the PacBio reads on their own. So we've done that now in microbes, yeast, rice, up to human, and get really good results. Although at contigs that are hundreds of kb megabases, the only gap though is if we have extremely deep PacBio coverage, so more than 50x coverage. There are some other algorithms that are really optimized for that, just expensive and to use.
It'd be okay even if the organism you're using doesn't have sufficient kind of like reference genome already.
Yeah. I mean, we do tests in genomes where we know the right answer just so we can check along the way. But the goal for all of this is to then extend it to the next genome where there's no reference available. Yeah. Other questions?
Yeah, my traditional question. What about transcriptome analysis?
Yeah, so the concepts are very similar. The actual software that you're going to use, the actual hands-on, will be quite different. So this, like I kind of sort of hopefully explained, was that genome assemblers use coverage as a really important clue to determine where repeats are versus what is unique, what is reliable, what is not reliable. But in a transcriptome, your expectations on coverage are just so different, right? Because of this wide dynamic range and what's available. Such that in practice, yes, they all use the Bruijn graphs to assemble short reads, but then everything that happens next will be very different for de novo transcriptome versus de novo genome assembler. The package that I know and really like for de novo transcriptome assembly is called Trinity. It was published in Nature Biotech 2 years or so ago. In our hands, it works really well. That would be my recommendation if you're interested to do that. Other questions? All right, well, thank you very much for your time.